Hello everyone. Now we're going to look into the lecture on nucleic acid. We will then look at other lectures in terms of the other biomolecule or macromolecule. We're going to look into lipid, carbohydrate, as well as protein. We look at their structures, their monomers, the functional groups, and also what function they play role in. And only then we look at their metabolic pathway later on. In terms of this slide that is on the nucleotide, you can see here the nucleotide, which happens to be the monomer used to build either RNA or DNA. RNA is a single strand polynucleotide, while DNA is a double-stranded polynucleotide. And then in terms of their nucleotide, there's only very little differences between the sugar as well as the base. But in terms of the phosphate, it is same in terms of RNA as well as DNA. So just to recap, the phosphate will be similar, either it's a RNA or DNA. But in terms of the sugar, you will see here the OH at C2 prime. If it's OH, then it is ribose sugar. But if the OH is replaced with hydrogen, then it's a deoxyribose sugar. So that's the difference in terms of the sugar. There's also differences in terms of the bases here. You will have either purine or pyrimidine. Purine is a double ring structure, while pyrimidine is a single ring structure. And in terms of the bases, there will be 4 bases for RNA, while 4 bases as well for DNA. The bases for RNA could be A, U, G, C. Adenine, uracil, guanosine, and then cytosine. While the basis for DNA cannot have uracil, but it's more like ATGC, adenosine, or sometimes adenine, they call it. And then you have thymidine or thymidicine, and then you have guanosine and also cytosine, just like what is found in the RNA. The only difference is in RNA you have uracil, while in the DNA you have thymidine. Now we look at the nucleotide, function of the nucleotide, and then in a while we look at the roles of nucleic acid. Difference between nucleotide and nucleic acid. Nucleotide, as you know, is the monomer that builds the nucleic acid. So the nucleotide, the question arises, does it have any role other than just making into DNA and RNA? So that's the first function, that is building blocks of nucleic acid, that is RNA or DNA. So they are the monomers. It's just like amino acid. You have 20 different amino acid can be put together to build protein. So the monomer of protein is amino acid. Then the second sentence there, act as energy currency. You see this adenosine triphosphate. The adenosine is actually can be referred as a nucleotide, and ATP is the energy currency that is required in your body, in your cell, and very much required to do work, because the hydrolysis of ATP will release something like 30 to 32 kilojoule of energy, which is trapped in the bonds of ADP and inorganic phosphate. So when they are bonded together, they become ATP. Then when they are hydrolyzed, that energy will be released, and this energy can be used in order to do work. Okay. This ATP can also exist in the body as CTP, GTP, as well as UTP. These are all the nucleotide the triphosphate nucleotide, but the one that is referred as energy currency is ATP. In fact, these other triphosphate, CTP, UTP, GTP, in order to convert them to ATP does not require loss of energy. That means 1 ATP equal to 1 CTP. Okay? And then you have allosteric effectors. The nucleotide in our body also can act as to activate the enzyme. Enzyme got active site. They also have close to the active site something referred as regulatory site. So these NAD, ADP, what else? GDP, they can bind to this regulatory site and actually control the function of the enzyme. That is done by allosterically. Of course, they also have another way to control the enzyme, that is covalent modification. But this role of nucleotide plays in a role of controlling the enzyme, either activate or inactivate, by having this coenzyme binding to the regulatory site. Another role of nucleotide is in the building of the coenzyme itself, such as this, that is NAD, nicotinamide, adenine dinucleotide. You also have ADP, that is the adenine or adenosine. Then you also have FAD, all these are coenzyme. Flavin, adenosine, diphosphate, and you also have another one, NADP. All these are coenzyme, which the A part is actually built by the nucleotide. So the nucleotide become the precursor, the starting material in making the coenzyme, or to put it in a bigger picture, they call it cofactor. Cofactor is all the coenzyme as well as prosthetic group. So that was on the nucleotide. So once you put together the nucleotide, then you get DNA as well as RNA. Of course, you already know that DNA, they have genes in it. So from the particular genes or few genes can then synthesize protein. Or the whole genome itself, all the say 23 chromosomes that is found in your body need to be transferred to the next generation. So it is to ensure transfer of genetic information from one generation to the next, or even if there's a new cell is made, division of cell will have same number of genetic information which is found in a DNA. Okay? And then there's something here written about promoter. That's where the starting of the gene being read. So wherever there's a promoter, that's where the gene, reading will happen, and therefore the information will be accurately made into a protein. Okay, and then you have, in terms of mRNA, got three types: ribosomal RNA, messenger RNA, as well as transfer RNA. In short, they call it rRNA, mRNA, tRNA. We will look into detail about their function later on, but for now, you just want to know that the role of nucleic acid is in the making of DNA as well as RNA. Here given one other function which can be said to be unique, because maybe it's not a new finding. It's already found, discovered some 25 years ago. Basically, they thought the function of enzyme or catalyst or biological catalyst is solely made from protein. However, some time ago, they discovered that no, all proteins are not biological catalysts. In fact, there's an exception to it, and this is the exception where even nucleic acid can function like a catalyst or biocatalyst in the body, where here you can see ribosome. It is a nucleic acid. It can actually cleave the RNA just behaving like a enzyme that is made of protein. Therefore, we can generalize that most enzymes are protein, with the exception of some nucleic acid can also act as biological catalysts. So now we revisit again the structure of nucleotide. Earlier already spoken to you about the structure of nucleotide, but again here you can see that the phosphate is the one that, same for every nucleotide. Doesn't matter found in RNA or DNA. Then you have the sugar, which is a pentose sugar. You have also exose sugar, but the one for DNA, RNA, it has to be a pentose sugar. And in terms of pentose, it could be either ribose or deoxyribose. When the OH is removed, then it's and added with hydrogen, then it's a deoxyribose. If the OH is present as shown here, then it is a ribose, and it always comes as C2 prime. And also in terms of the purine as well as pyrimidine base, you have all five that comes into these two groups, that is A, U, T, G, C. Okay? And they vary in terms of only one being different for DNA and RNA. One will have uracil and the other will have thymidine. Okay, that's the only difference. Okay. Then you have also this term, there's no nucleotide, then you have nucleosides. Okay. Nucleosides, the SIDES, refers to, in chemistry, refers to anything that has sugar bound to it. But when it comes to this nucleoside compared to the nucleotide, you have the phosphate gone missing. So that's why it's called nucleoside, not nucleotide. And what's the function of having the phosphate not existing in a nucleoside is still not clearly understood. But just to say that if there's no phosphate, then it's nucleoside, and if there is phosphate, then it's a nucleotide. Then we look at the sugar itself, the ribose. It can be, as you can see here, it can be acyclic or cyclic. And then it is furanose. Furanose means 5-membered ring. Okay? You can see it's not 5-carbon ring, but 5-membered ring, which has carbon as well as oxygen. So that's furanose. And in terms of sugar, which is like hexose and also galactose, they are actually pyranose ring structure. Okay. Bit more in terms of why the prime comes in. This prime is actually, as you can see here, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay, it's just labeling based on chemistry way of referring to a structure. For the sugar in the nucleotide, it will be marked as 1 prime, 2 prime. While the base, which is attached to the sugar, the bases A, T, G, C, and U, their members are labeled as 1, 2, 3, 4, 5. Just to differentiate between the sugar as well as the base labeling. Okay, this slide is detailing out the deoxyribose. Okay, if the OH here is removed and put with hydrogen, then it is called deoxyribose. Okay. And then deoxyribose is in DNA, while ribose is in RNA. If you put the OH here, then it's a ribose. Then it also referred here, the sugar prefers a different puckers in DNA as well as RNA. Puccus referring to the C2 prime endo. What is C2 prime endo? Which is normally found in DNA, but in DNA also have C3 endo. Similarly, in RNA mostly will be in C3 endo, but there's also C2 prime endo. Okay, so what is this C2 prime endo and what is C3 prime endo? The C2 prime endo referring to the carbon 2 prime being closest to the C5 prime carbon. So that's why it's tilted closer to the C5 prime, and therefore it is referred as C2 prime endo. If you have the C3 prime carbon to be close to the C5 prime, then it's referred as C3 prime endo, and that's normally found in RNA. So that's the whole difference in terms of C2 prime endo as well as C3 prime endo. So this slide is just to show the difference in structure of pyrimidine as well as purine. Okay. So you can see pyrimidine has one ring structure, purine has two ring structures. And you can also know where the connection of the sugar will take place in the pyrimidine as well as in purine. For the pyrimidine, the connection of the ribose or deoxyribose sugar will be at N1 location, while for the purine will be at N9 location. Okay. And in terms of the pyrimidine and purine, you have all the five bases to be part of these two groups. Now we look at the bases that belongs to purine as well as the bases belong to the pyrimidine. As you can see on the top left, you have adenine, guanine, and on the bottom right, you have cytosine, thymidine, as well as uracil. In terms of the thymidine and uracil, thymidine only found in DNA, as well as the three other bases, that is adenine, guanine, cytosine. While in RNA, there's no thymidine, but you have adenine, guanine, cytosine, as well as uracil. So that's the understanding about this slide. Next, we have more understanding about the nucleotide structure, where the phosphate group found in one nucleotide, and then it comes together to become polynucleotide. Therefore, these phosphate negative charges play a major role. And this phosphate group having the negative charges will then attract positive charges, the counter ions, such as positive charges such as from magnesium. Magnesium is the cofactor for DNA, RNA, and also this method they refer as PCR, polymerase chain reaction, which you will learn in genetics or gene technology. There you will see you need to have this cofactor magnesium when you want to do this PCR technique. And you also have the polyamine. Polyamines are also positively charged, and they will be attracted to the, say, DNA having these phosphate groups. Okay. And also you have the protein histone. This histone combines with this negative charge, and therefore the chromatid, you remember your genetic lessons, will become chromosomes when the histones are bound to this negatively charged phosphate. And in terms of the other facts, you also have the mentioned here bases attached to the C1 or ribose of ribose or deoxyribose. Okay, so the C1 prime will be the location the bases will bound, and in terms of the bases, as I already showed you, the pyrimidine at N1 position while the purine at N9 position. So here we're going to focus our attention towards the deoxyribonucleoside, then about deoxyribonucleotide. Basically the difference is one has phosphate and the other one do not have phosphate. Okay? So you have firstly it is called deoxy because there's no OH, instead there's hydrogen at the sugar. Then you have the phosphate, not there, therefore it's called deoxyribonucleoside. And then when the phosphate comes at the position of carbon 5 attached to it, then it will be referred as deoxyribonucleotides. Okay? And the next statement is referring into how to abbreviate these deoxyribonucleotides. It can be simply called as A or DA or DAMP or deoxyadenosine monophosphate. So this is the abbreviation, but easy just to refer them as A. Here, given all the four major deoxyribonucleotide, you will not see deoxyuracil, because it's not existing in DNA or deoxyribonucleotides. So you have the first one, deoxyadenosine, simply referred as A, or the nucleotide deoxyadenylate itself, referred as that. Then you have nucleoside in the form of deoxyguanosine or deoxyguanylate, and then you have deoxythymidylate or the nucleoside as deoxythymidine, and lastly you have deoxycytidylate or deoxycytidine. Interesting to see that if it's a nucleotide, it's referred as, for instance, deoxyadenylate. If it's a nucleoside, it's referred as deoxyadenosine alone. So similarly for ribonucleoside as well as ribonucleotides, so you have the phosphate. If it's taken out, then it is referred as ribonucleoside. Then when you add the phosphate, it is referred as ribonucleotides. And then in terms of how it's been abbreviated, it could be simple as U or UMP. Surprisingly, not the Universiti Malaysia Pahang, but UMP as in uridine monophosphate. Here for the ribonucleotide, you have the four major ribonucleotides shown. So again, being separated as nucleotide, symbol as well as nucleoside. So again, if you look at it carefully, there is no presence of thiamidylate. There is no T in ribonucleotides. Okay, so you have adenosine, guanosine, uridine, cytidine only for ribonucleotide. So here shown side by side, on the left DNA, but only single strand being shown. By right, it should be having two strands. It's a what you call double-stranded DNA. One strand runs from 5' to 3', and there's another strand which is not been shown here from 3' to 5'. I'll explain to you in a while what is the meaning of 5' as well as 3' N. Okay. And then you have on the right RNA, which is a single strand alone, and you have here also explanation on phosphodiester linkage. The phosphodiester linkage comes in between here. Yeah, these two locations here, which involves the phosphate as well as the CH2OH of the neighboring nucleotide. So when the phosphodiester linkage takes place, there will be removal of water molecule, or referred as condensation reaction. Okay? So that is phosphodiester linkage, which happen to be the covalent bond of nucleic acid. Okay? The 3D structure is actually contributed by the hydrogen bond, which in a while we look at it as well. So just now I say 5' position as well as 3' location. The 5' and 3' easily can be understood by counting the location of the members, starting with C1', C2', C3, C4, and C5. If it ends up at C5' at one end of it, therefore it is referred as strand beginning at C5' end. And when you look at the bottom, at the far end, lower end of the polynucleotide, either the DNA strand or RNA, you then count C1', C2', and C3'. It's already reaching at the end of the strand. Therefore, that is C3' position end. So that's how you understand the C5' end as well as C3' end, which happen to be found in both DNA as well as RNA. So more on the what we call base sequence of DNA primarily. So here shown in terms of something referred as sugar-phosphate, sugar-phosphate. It repeats in this manner, and therefore it's called sugar-phosphate backbone. Okay, and then also been highlighted that the DNA is puckered in C2' endo. I already shown what's the meaning of C2' endo. The DNA also can be said having nucleotide built by nucleotides, just like amino acid building protein. And there is also ways to write the sequence, and it always starts from C5' N and until the base that is found in the C3' N. Okay, so that means the C5' is on the left. So just to find if it's A there, so adenosine. Then the next one is thymidine, and then guanosine and cytosine. So you write it as 5' N, ATGC, and so on if it's longer. The last note there referring to the backbone to be polyanionic because of the phosphate charges. Therefore, it is anionic or anion or negatively charged, which attracts the positive charge counterions. And now we come to the 6 main features of the double helix model of DNA. Okay, at least 6 major features. So the first one, I just go through the facts, and then next slide I show you in the model itself. So the first feature is 2 polynucleotide chain wound in right-handed clockwise double helix. If you were wearing a watch which has needles, therefore the clockwise is referred as the needle going from 12 to 6 and then 6 to back to 12. So that's clockwise. Okay. So in terms of this DNA polynucleotide, the facts say it is clockwise. So if you put your hand, okay, your hand like this, then you grip it. Okay. If the hand fingers here turn like a spiral on the DNA going up, you will notice the spiral turning is in fact clockwise. Okay, but if you take the left hand, the spiral will go anti-clockwise. So for DNA, double helix is always rotating right-handed manner and is also referred as clockwise. I know it's a bit difficult to understand. You try to find some YouTube videos on it, or go and find Google it, and you will understand better. Okay. And then you have the second facts. Nucleotide chains are anti-parallel. Okay? So you have the one, double-stranded DNA. One strand is 5' to 3' end, and another one next to the 5' will be actually 3' beginning, and then finally will end as 5'. So they are referred as double-stranded anti-parallel positions. Okay? The third one is the sugar phosphate backbone are on the outside while the bases are inside. This is probably to protect the bases because the bases will be the one that decides on the gene, the codon, the anti-codon. So they are put inserted inside to be protected, and the sugar phosphate backbone will be on the outer side. And also the counter ions can approach the phosphate bed much better if they are outside. Okay. The fourth one, complementary base pairing. Okay. So they call it as Chagaf rule, where A base always go with T and then G with C. And A and T have two hydrogen non-covalent bond, while G and C have three non-covalent hydrogen bond. Okay? So this is Chagaf rule that defines it has to be complementary, A to T, G to C. In fact, A also go with uracil, if it's DNA with the mRNA. Okay, and the fifth facts refers as base pairs are 0.34 nanometers apart. Each nanometer is 10 power of negative 9. Okay, so got 10 base pairs. I'll show in a while. The length or the distance between the first and the 10 is 0.3 nm. The first and two base pairs are only 0.34, while number 1 until 10 will be 3.4 nm. So that encompasses 10 base pairs. Okay? I'll show you in a while to understand better. Okay? And the last fact is about the sugar phosphate backbone. It's not having same spacing. One will be bigger space and another one shorter spacing. Therefore, it's referred as major as well as minor grooves. Here we can explain about the 6 features. Basically, you can see the double-stranded, and then you have the A complementary to T, G with C, and then G and C got 3 hydrogen bond, A to T got only 2 hydrogen bond. And then also you can see one strand is 5' to 3', not very clear here. Another strand is anti-parallel, that means 3' to 5'. And then you can see the minor groove as well as major groove. Also shown the 3.5 nm, which will be a total or encompassing 10 base pairs. And then between one neighboring base pair to the other is only 0.34 nm or nanometer. Okay. I think covered most of the 6 features. So more on the 6 features been shown in a schematic diagram. You can see there in terms of the adenine, the structure linking up to thymine with 2 hydrogen bond. You can even see the length between the bonds between A and T. You also have guanine linking up to cytosine in the antiparallel strand of DNA with 3 hydrogen bonds. Okay. Um, and also on the left, you can see what has been missed out in the earlier slide about the 5' to 3', and then the anti-parallel strand of 3' to 5'. This is interesting in terms of also already been taught to you in earlier slide about complementary but not identical. If it's identical, instead of A complement to T, G with C, it should be, if it's G, it will link up identically to G, or A with A, or T with T, or C with C. So it doesn't work that way, but rather because of the Chagaf rule, it is complementary. Which also means if I were to give you a sequence of the one strand of the DNA, it is possible for you to find out what's on the other strand, the sequence itself, by looking at the sequence on the strand that is been given to you. From DNA, we now look into the structure of RNA. RNA basically involves 3 types, rRNA, tRNA, as well as mRNA. And here shown the structure of tRNA. Interestingly also, you can see that in tRNA that is been shown there, there is no thymidine, only uracil is been shown. So in line with the DNA complementary pairing, whereby A is always paired with T and G with C, here you can see at the top left corner, A pairs with U and C pairs with G when it comes to discussing about RNA. Okay. And then divided to mRNA, tRNA, RNA. M stands for messenger RNA, T for transfer RNA, and then R for ribosomal RNA, or ribosome RNA. Okay. In the middle there, shown the yeast alanine tRNA, and shown that the mRNA at the bottom has this codon which sticks, or complementary binds to the anticodon found on the tRNA, and this brings about the amino acid within the tRNA to be building into becoming a polypeptide. Okay, and this particular tRNA, if you look at the 3' end, there is the amino acid alanine attached to it. So the next few slides discuss about each one of the mRNA, beginning with tRNA here. It is the smallest, and its function is to transport amino acid. So it has the anticodon with the tRNA, while the mRNA has the codon, so they are complementary to each other. It is understood for the 20 amino acid, they could have more than one particular tRNA that is used to anchor the 20 types of amino acid, meaning there could be 2 tRNA what you call responsible for one amino acid. Next is on mRNA, or messenger RNA. You can see it is degraded after use, meaning it is made in the nucleus after complementary build-up from one strand of the DNA, and then it goes in out of the nucleus into the cytoplasma, looking for either free ribosome or ribosome that is attached to endoplasmic reticulum. And after that, once the protein is built up or polypeptide is built up, the mRNA is degraded. Okay, so that's the meaning of degraded after use. And the new mRNA form basically just to produce a new protein. Its function is just for that. And as I said, it is made in the nucleus and it actually travels to the ribosome in the cytoplasma. So it's mobile. What I can tell you, why is it mobile, is because it has, apart from the sequence of nucleotide made, it also attaches to itself adenine, polyadenine tail, and this tail is used to swim out the mRNA from nucleus to cytoplasma. The third of the type of RNA is ribosomal RNA, and this one is in short is called rRNA. And this rRNA will be used to make a bigger component, and that's referred as ribosome. And this one is the place where the mRNA will be anchored, and therefore it is used in building polypeptides. So here given a bit of the function of the three RNAs, mRNA, rRNA, as well as tRNA, where it is used to generate the polypeptide, whereby you can see the ribosome is where the mRNA which been made in a correct sequence of nucleotides, and there you have the codon in it. This codon will be specifically bound by anticodon, where the tRNA comes with an amino acid, and once it binds to the codon of the mRNA, then the peptide bond will be made between the what we call increasing polypeptide, and this goes on until it reaches the end, or stop codon they call it. So we have come to the end of the this lecture, and normally at the end I will have a student activity, and this is supposedly should be the student activity for nucleic acid if it was done in a classroom. So do go through the YouTube that has been given here relevant to nucleic acid, and then to understand this lecture better, you ask this question and see whether you can find out the answer for it. Thank you so much.